Okay, thank you very much, Deborah, for the introduction, for the invitation to speak here. I think this is the only really scientific activity that I have during these 3 days. I'm looking forward to it, other than just having pictures taken and stuff, and meeting kings and presidents, which is also great. Anyways, so yeah, I wanted to tell you a bit about latest developments in my lab, and in the context of what I call synthetic mechanobiology, and specifically about shape, about migration, and about cancer. So let me begin with I would say almost philosophical introduction. I would argue that there are 2 main ways in which you can understand a living system. And the first one is through these top-down approaches. The way this works is the following. So you identify a process. And it can be your favorite process, a context of cell migration, or a developmental process, a specific biological process in disease. You pick your favorite biological process. And then what you do is you measure everything you can in this system, right? And today we have wonderful microscopes. We have all possible omics that you can imagine, so we can really measure lots of things, right? And then you model the system, and then crucially, you deconstruct it. And by that I mean you remove one essential piece of the system so that the system no longer works as it should. And then you go back to measure everything, to model everything, and again you deconstruct. So you learn how the system works by deconstructing it, right? So this crucial part of you knock out one gene or you overexpress it, but basically you break the system. Okay, so this is extremely powerful, and I would argue that most of what we know about many systems comes through these top-down approaches. Then there's an alternative, which is bottom-up approaches. So here, instead of learning by deconstructing or learning by breaking, you learn by building. So you try to put together the elementary building blocks of a system, and you try to assemble them so that the system works as it works in nature, right? So this is called bottom-up approaches, and I call this learning by building. So I'm not here to say that one is better than the other. I think many of us use both of them. But in my lab, over the years, we have sort of embraced these bottom-up approaches. And this is what I will be telling you today about. So in fact, we wrote about this already in the past. So there was one issue, in fact, some time ago already about this idea of bottom-up biology. We wrote one paper in this issue. We've written other papers more recently, also specifically context of mechanobiology, which is this interplay between the world of mechanics and the world of biology, and others have also written some very nice studies on this. So if you're interested, there's already some literature into how these systems are actually useful in research. Okay, so in the lab we have 3 main lines where we apply these ideas. So the first one is to use bottom-up approaches to study tissue migration. Another line is to study shape. Another one is to study cancer. So the main person who has developed this line of research in the lab has been Anna. And I'm sure that she has already told you about a lot of this, so I will not touch cancer today. I will focus on these 2 lines of research, bottom-up tissue migration and bottom-up shape. And mostly I will be telling you about migration. Okay, so I'd like to start my talks about migration with this movie. This is a movie that is already quite old, more than 10 years ago. And basically what you're seeing here is a classical experiment of epithelial wound healing. So you take a monolayer of epithelial cells, you scratch it either with the tip of a pipette or, as we did here, with a laser, and then you just watch what happens. And what happens is that these cells start crawling in order to close the gap within about 1 hour. This is a classic experiment. It has been done many years ago. Any master student can do it. And so a long time ago, we started thinking about this problem, and we realized that we were not able to understand it unless we understood the forces that drive this process, right? So I can give you every possible omics measurement of the system. I can give you the state of every protein, of every gene in this system. You will not be able to tell me how this system is closing, right? Because these cells are moving. Moving is a problem of mechanics, and mechanics needs physical force. So we were very surprised that we didn't have tools in order to measure the forces that these cells are generating in order to close this wound. So we spent a lot of efforts in developing these tools, and I will not tell you today a lot about technologies. We wrote a paper that's sort of a review of all the technologies that we and others have developed in this context. I will tell you a bit about this technology that is called traction microscopy. The idea is actually quite simple. You have a group of cells that are migrating in this direction. As these cells migrate, they generate forces on their underlying substrate. These forces are very small. They are on the order of nanonewtons, so very, very small. So the idea is that if you make a substrate that is very soft, like a hydrogel, and you insert beads inside the substrate, as these cells are crawling, these forces will deform the substrate. If we measure this deformation with these fluorescent beads, then we can solve a problem in continuum mechanics that allows us to know how much force every cell is exerting at every point of the interface between the cell and the substrate. So this traction microscopy is the main technology that I'll be telling you today about, of all the sort of arsenal of techniques that we now have in the lab. So thanks to this technology, we could turn this movie into this movie, where we now, at every point of this movie, we can now put a vector, which is a force vector. It's indicating how much force a cell is exerting on the substrate. The movie is color-coded according to the radial component of the forces. So red means that forces are pointing away from the wound. Green means that forces are pointing towards the wound. And if you pay close attention to this movie, you will see that at the very beginning of the movie, most forces at the edge are red, whereas at the end, most forces are green. So this corresponds to two different mechanisms. Red forces corresponds to cell crawling. So these are cells that are grabbing on the substrate, and they are propelling their body forward like this. So the force is actually pointing towards the cell, away from the wound. So these are the forces that are associated with cell migration. By contrast, and the green forces, they are contractile forces. They are generated by this accumulation of actin lining this gap. And this accumulation of actin and myosin leads to a contractility that drags the monolayer and results in forces that are pointing towards the gap. Okay? So then once we had these forces, we were able to develop computational models in which we add these ingredients, we add these lamellipodia, we add this ring, and then we could match one-on-one the model and the data. And this really allowed us to understand what mechanisms are at play, to what extent they are at play, when they come into play. Okay? And only through these mechanisms one can actually understand this problem. And now, of course, we can ask questions of what can we tune the system to improve these mechanisms? Can we understand the molecular mechanisms that are behind force generation and so on? Right? Okay. So since then we have worked on more and more complex systems where we apply these technologies. One of these problems is the intestinal epithelium. So the intestinal epithelium is shaped with these finger-like protrusions that are called the villi, and that they serve to essentially pack a very large surface into a small volume to sort of maximize the intake of nutrients and fluids. And essentially, the intestinal epithelium has sort of this incredible property, I would say, which is the fastest self-renewing tissue in our body. So the cells that line your intestine are renewed every week. So every week, the entire surface of your gut is new. And this happens thanks to stem cells that are located at the base of these invaginations that are called the crypts. So these cells divide, then they move along this transit amplifying zone. They continue to crawl all the way up to the villus, and eventually, as they reach the villus, these differentiated cells, then they are extruded. Right? So there's this very rich biological process in which these cells must regulate stemness, compartmentalization, folding, then they proliferate, they migrate, they differentiate as they migrate, and then they are extruded. Right? So in the lab, we're interested in how these different functions are coordinated. So how every cell knows what function it has to perform at a given time, at a given location, to what extent this coordination might be mechanical in origin. So for that, these two people, Carlos and Gerardo, PhD students in the lab who have now moved on to their postdocs in collaboration with Daniela and Marino Arroyo, we developed this technology so that we can now use intestinal organoids, and we can now not just visualize the system. So here are where the intestinal stem cells are. These are the proliferative cells. These are the differentiated cells. You can, if you good attention, you'll be able to see that some of them are actually being extruded. So we can now visualize this process of intestinal homeostasis under the microscope, and we can actually also measure the velocities of the cells, which is what you see here, and we can measure the forces that these cells are exerting. So this is very rich. I could spend my entire talk just discussing these movies. In fact, I could spend my entire career just looking at these movies, which is an option, but I will not do that. I just wanted to show you, to make the point that we can now use these technologies and push them so that we can really apply them to systems that are not just simple epithelial, but that are actually simple, much more complex systems. So one part of my lab is now sort of moving towards looking at things that are increasingly more complicated, and the other part of the lab is moving in the opposite direction and looking at systems that are increasingly simple. And today I will be telling you about that. And the first problem that I would like to discuss in this context is the problem of leaders and followers in collective cell migration. So if you take a group of cells and epithelial cells and you put them on a dish, it's quite likely that you will observe something that looks like this. So these cells will get together. They will migrate collectively. They will form these strands. And if you look at the tip of these strands, you will often see a cell that is larger, that is more protrusive, and that seems to be playing the role of a leader. And this type of configuration, where you have a cell that seems to be a leader and then some cells that seem to be followers, this is some sort of organization that you see often in biological systems, both in physiology, development, and disease. So this is one example in the lab. This is work by Anna when she was interested in understanding the relationship with fibroblasts that you see here in red and cancer cells, and she was able to discover that fibroblasts are actually pulling cancer cells away from tumors in one configuration that seems to be one of these leader-follower configurations, where now the calf is a different cell type, is actually, what the leader is a different cell type, which is the calf. In development, this is the development of the lateral line primordium in zebrafish. And this system is also migrating in one dimension. You see this cell that is at the tip seems to be more protrusive, extending more protrusions that seem to sort of be leading the migration of the remaining cells. So this is a type of configuration that is actually quite prevalent in biology, and this has led many authors, many scientists to think for generations about how are crowds mechanically organized to move together. And there are essentially two big schools of thought here. One of them is those who think that migration is essentially organized in a way that there is one leader that is exerting all the guidance in the system, and then there are passive followers that will just follow this leader. And then there's another school of thought that thinks that the system is somewhat self-organized, so that every cell is in a way contributing to guidance. And in fact, this is an interesting discussion because it always ends up being a political discussion between those who actually think that all we need is very strong leaders who want to make the monolayer great again, versus some sort of others who think that everyone should contribute to the common good of the monolayer. So, so politics aside, when you think about this problem in terms of physics, and this comes down to finding a force-velocity relationship. And force-velocity relationships, which is sort of a fundamental of mechanics, is a problem that we have solved for pretty much any system that exists in nature. So we know force-velocity relationships for black holes, for galaxies, we know it for atoms, we know it for quantum mechanics. But we don't know it for cell migration. So we don't know how much force each cell here needs to exert for the system to move in a specific way. We don't know, okay? And this is what we wanted to find out. And here's where Leone Rossetti comes into play, postdoc in the lab, who now runs his own lab at King's College in London. And with Leone, we started thinking about, can we design a system that is simple enough that will allow us to understand this problem of leadership? And after thinking a bit about it, we came up that this is the experiment that we wanted to do. Okay? So we want to do a system of collective migration, as you see here. We want a small number of elements in the system. We want them to move in a simple geometry, so one dimension is better than 2 dimensions. And we want to be able to add a leader to the group at will, on demand. So we want to add the leader, we want to remove the leader, and so on. So we don't know how to do this experiment with penguins. Maybe your colleagues here at the zoo can do it, because they are really amazing scientists. I was there yesterday. They were talking to us about the wonderful science they are doing. But anyways, we don't know how to do this in penguins. But Leone found out how to do this with cells. So first thing you need is to generate leaders, right? And so the way you can generate a leader is through optogenetics. So we developed this line that is sort of a photoactivatable cell line, in which when we shine light to one region of the cell, we trigger the formation of one lamellipodium, of one protrusion. And the way we do that is by localizing a RAG1 GEF that upon illumination localizes at the membrane of the cells and then activates this process of protrusion. Okay? So this works very well, and that's something that Leone was able to develop. When you stop shining light, then this protrusion decreases. So we can turn a cell into a cell that migrates. Okay, so that's the first thing we needed. We can turn a cell into a motile one that can act as a leader. We also want these cells to migrate in 1 dimension. This we also know how to do. So we just print lines of extracellular matrix on the substrate, and then we sit cell on top of that, and then we can have cells migrating in 1D. And there's other people who have sort of done this before us, so we're really like late to this party. But anyway, so these are raw data. So these are the raw data that Leone did. So you can see that cells are migrating in 1 dimension. You have clusters of different sizes. So like quadruplets here, you have a single cell here, you have doublets. And these blue squares, this is Leone shining blue light, very, very dim blue light, on one of the edges of these cells. And this works as intended, right? Pay attention to this doublet here. You see how there is light being shined on one half of the cell, and then immediately the cluster starts migrating in the direction in which we are sort of intending to move it, right? And this happens here as well, happens here as well. In other clusters, this doesn't work at all. I mean, you look at this one, Leone shines light here, and the cell moves in the other direction. They couldn't care less about illumination, right? So there's some variability in the system. But the beauty of these sort of bottom-up approaches of using these reductionist systems is that they allow you to do many experiments and very systematically. So you can really do high statistics on this type of systems. And I'll be insisting a lot on this. So Leone now can analyze singlets, doublets, triplets, and quadruplets. And he can ask, for example, the simple question of whether cells, after photoactivation, are moving in the direction in which we are photoactivating them. And this is shown here. So this is the position of the center of mass of these cells for cells that are activated, for cells that are not photoactivated. Positive here means that these cells are actually moving in the direction where we are photoactivating. So for single cells, most of these cells are actually migrating in the direction that we want them to move. So that's nice. How about doublets? For doublets, the same thing happens, right? So we photoactivate cells, and most of them, they move in the direction of photoactivation. So basically, in this arrangement, we are able to generate one leader that is actually very good at leading one follower. So that's very good. When we go to bigger groups, like triplets and quadruplets, this effect is gone. So there's no difference between these groups or between these groups and 0. So basically, for groups bigger than 2, this autonomous leader that we are generating is no longer able to lead the migration of the followers. So what this tells us is that this idea that just one cell that is an autonomous leader, that is big, protrusive, and so on, is able to lead the migration of a large group, this is not sustainable. Right? We could make better leaders, maybe, but we will not be able to generate leaders that lead many, many cells. Right? So this sort of, in a way, rules out this vision. But then what Leone wanted is to really understand why, right? So why we can lead just one follower and then not bigger groups. So what does it take for a cell to lead a group of cells? And for that, we turned again to physics, and we turned again to force measurements. So this is again raw data. Here's the experiment. Two cells are photoactivated here and there, and then as soon as they're photoactivated, they migrate in the direction where we want them to migrate. And these are force maps. So these are the maps of these tractions that cells are exerting on their substrate. So this is color-coded according to the sign, and the sign is shown here, right? So either you're putting away from the leading edge or towards the leading edge. So as you can see, there are some hotspots here, so this is not a completely trivial force field, but again, we have many of these as a function of time, so then we can do statistics. So Leone looked at many of these groups, and what he found is that when you have a single cell, basically what you have is two hotspots of traction, one at one edge and the other at the other edge. So basically what this cell is doing, it is grabbing on the substrate here, there. It's contracting. Okay, so this is what these force maps are telling you. And for doublets, you get the same. As you go to bigger groups, then you start seeing that there are hot spots in the middle. Okay. So then Leone said, well, I mean, this is just one example. Let's average them out over time and over many experimental repeats. And he did that first for the cells that do not move. Okay, for cells that are either not photoactivated or cells that are photoactivated and do not move. And when he did that, this is what he found. So these are distraction fields as a function of the normalized length. So this is one edge, this is the other edge, so this is here, so this peak is this peak, and this peak is this peak. Okay? So basically what you find is a symmetric distribution of force with a force peak here and a force peak here. And as you go to bigger groups, you see the same thing. You see that basically there's nothing on average in the center and just two peaks at the edge. Now, if you go to the cells that do migrate collectively, here's what you find. So instead of finding a symmetric system, you have now an asymmetric distribution. The magnitude doesn't matter. So if you're very strong or not, that will not make you migrate faster. And this was a misconception that is in the field and that we also had. Right? So more force does not necessarily mean more motion. What means more motion is the asymmetry in the system. So the cells that are moving have a peak that is similar at the leading edge, but at the trailing edge they have a distribution that is actually much lower. Right? And this happens for single cells, doublets, triplets, and quadruplets. So all this data pointed to this idea that what matters is the asymmetry in the system. I showed you cells migrating in one line. You might object that cells do not migrate in one line. I would say in some cases they do, in others they don't. So as requests by referees, we tried many other systems like monolayers, like clusters of cells, and we found the same, that the cells that migrate, they have this asymmetric distribution of traction forces. So we decided to measure this asymmetry. There's different ways in which you can measure an asymmetry. The one that we picked is this normalized quadrupole. So if this doesn't tell you anything, which it probably doesn't, just take this as an indicator of asymmetry normalized by length and by magnitude. So if Q is big, then asymmetry is big. And this has a sign. You can be asymmetric in one direction or in the other direction. Okay? So then Leone computed, or plotted, in fact, the average velocity of each one of the clusters he measured as a function of this asymmetry. And what he found is that either for singlets, doublets, triplets, and quadruplets, there was a relationship that was telling us that the higher the asymmetry, the faster these cells were migrating. So this established, for the first time, a relationship, a physical relationship that explains or that describes what a cell needs to do in order to move fast. So what you need to do is to develop a force distribution that is asymmetric. So as the conclusion of this, there's 2 points. The first one is that leaders are inefficient at driving collective cell migration. And the second one is that the traction asymmetry along the cluster is what determines cell velocity. And notice that this does not solve the problem between this distribution and this distribution. What we're saying is that you need an asymmetry, which you can reach in this way, or you can reach in this other way. But without this mechanical asymmetry, you're not going to move. Okay? So that's the first point that I wanted to tell you about today. Okay, so the second part, we stick with cell migration, but in a very different context. So instead of telling you about collective migration, which is what in the lab we have been studying systematically for many years, I will tell you about single cell migration, which is actually new to us, in the context of gradients. So you're probably all aware that one way in which cells can migrate in one specific direction is through gradients in their environment. So if the gradient is in a sort of a soluble chemical, this is called chemotaxis. If there is a gradient in the substrate stiffness, then this is called durotaxis. And there is a gradient in the density of the protein of the extracellular matrix. This is called haptotaxis. There are other taxis. There's electrotaxis, which is becoming very popular, for example, which is gradients in electric fields, and so on. So Isabella, former PhD student in the lab, who's now doing her postdoc, was co-supervised with Raymond Sunyer, another former postdoc in my lab, in a collaboration on the theory side with David Bruckner and Eduardo Geneso. We decided to study single-cell haptotaxis. So what Isabella managed to do very carefully and very robustly is to print gradients of extracellular matrix, and specifically fibronectin. So what you see in red is fibronectin. And as you can appreciate, there is a gradient in fibronectin. So there's low density here, there's high density here. And she managed to print these gradients in fibronectin along specific lines. Okay, so there's no challenge. This is a flat surface in which you print a line of fibronectin that has a gradient. Okay, and then she sees cells on top of this line, and then as you would expect, cells move towards the highest concentration of fibronectin. We've known this since the 1960s, so no surprise here. We just found a fancier way to look at it, but essentially pretty much everyone who looked at this problem before us got it right. You can plot these trajectories in the following way. So this is x, so this is 0 means here, minus 250 microns means here, and this is time, so this trajectory moves upwards, right? Okay. In some of Isabella's experiments were actually more interesting because we had cells that are actually moving away from the gradient. And again, the nice thing of having such highly controlled experiments is that you can measure many, many of these cells and do very nice statistics. So Isabella asked how many of these cells move as everyone thinks they should, like apoptosis, and how many of them move against the gradient. And so first she did a control where there is no gradient, you just have one line of continuous ECM, a constant density of ECM, and then you have the 50-50 the cells move either towards the right or towards the left, which is what you would expect. Now you put a gradient, and then most of the cells, 83%, move up the gradient, 17%, which is not zero, actually move away from the gradient. Okay, so we try to understand this, and we understood this in terms of clutch models. So clutch models is one of the great successes, I would say, of biophysics in the past 10 years, because it's models that really, really allow us to understand how the leading edge of a cell works. And this is something that was developed originally by Chen and Ode in science, and it was then developed at our institute with Pera Roca-Cusacks and others. And we really now have a clear idea of how this works. So the idea is that at the leading edge of a cell, you have actin filaments, of course, they are protruding, and this protrusion actually pushes the membrane forward. But at the same time, you have contractility driven by myosin that is actually pulling filaments backwards. And this leads to a retrograde flow of actin. So even if you're polymerizing here, actin is actually moving backwards within the field of reference of the cell. And then this flow of actin is actually connected to the extracellular matrix through integrins and through adaptor proteins such as stalin. And as myosin pulls, then the substrate is deformed. Now these adaptors and these integrins have on and off rates, so they bind and unbind. And when you put all this together into a model, you can actually get a pretty comprehensive prediction of how the leading edge of a cell works. So in our case, this was a bit more complicated. We have two edges in a cell that is migrating. And you have sort of all these ingredients. You have them at one edge and at the other edge. Now, if you assume that there are more ECM ligands on one side than on the other side because there's a gradient, then you will have more integrins engaged on one side than on the other. And then when this system contracts, it will be easier to detach the trailing edge than the leading edge. So it's straightforward that the cell will move in this direction, right? So this is really, really a straightforward prediction. So all this sort of this modeling, all this data, everything seemed to make sense. But then Isabella did an experiment that is actually a much more clever experiment than the one that I asked her to do. So I've been telling you about this, but this is actually not the experiment that Isabella did. The experiment that Isabella did is this one. So she made sort of a mountain of fibronectin, a triangle. So a mountain is not a physical mountain, it's just that fibronectin increases, but then it decreases. So then she put a cell there, and that cell starts moving up the gradient. But then it reaches the point of highest fibronectin density. And this is where the interactive talk starts, because this is a quiz. This cell has three options. The first one is not to move. So you stop there, you're happy, highest density, I'm stopping there. The other option is continues to go, but then it has to continue down the gradient. And the other option is it turns and moves down the gradient, but in the other direction. Who thinks that the cell stops? You have to raise your hand. One over there, a few here. Don't be shy, don't be shy. You're on TV, but don't be shy. Who thinks that the cell continues to move? In this direction, good. In the other direction, okay. These are the disruptors. Okay, the solution is that the cell continues to migrate. Okay, and this is something that our model did not anticipate. Okay, our model would predict that once you hit the region of highest adhesion, that's where you get stuck. And we quantify this, so if the gradient is uniform, you have, again, half and half of them. But here, when you have this gradient, then some of them, to start with, go in that direction, but then the rest, they migrate. And once they hit the center, most of them continue to go. Okay? They don't continue to go at the same speed. You can see here that they're beginning to slow down. And in fact, if you compute the velocity, on average, you see that when they're going up the gradient, they are faster than they were going down the gradient. Eventually, of course, they will slow down and they will have zero. Okay? So this was challenging our model. Our model was not, and actually any other model was not standing. So with David and Eduard, we sort of pushed the model a bit further. And I will not go into these differential equations, but basically what we learned is that you need to take into account two elements, right? So the first element is gradient sensing, right? So adhesion of the cell. And the other one is actually the persistence of the cell, right? So this cell is polarized, and it has some sort of inertia that will make it continue to go no matter what, right? And so those of you who said the cell will stop is because in your mind you're sort of adhesion people, right? Maybe you don't know it, but you're adhesion people. So you think that adhesion will dominate migration. And the other of you probably are sort of more polarity people who think that polarity actually, once you're polarized in one direction, just keep going, right? So, okay. If you add these two ingredients and you sort of tune these sort of persistent random walks and so on, you end up being able to predict distributions that are very close to the ones that we observe experimentally. But then you now need to sort of take into account these two ingredients, right? So this was really good. It was very satisfying. It was time to write a paper. But whenever it is time to write a paper in my lab, we don't write the paper. We continue to do experiments. And that's because Isabella thought that, well, we think that what we're seeing is actually strongly dependent on confinement, and we have not touched confinement, right? So this cell cannot turn, right? So we sort of make it migrate in one narrow alley, so it cannot turn. What if we now offer that cell the possibility of turning? So we make a gradient that is like a 1D gradient on a 2D surface. Okay, and now we put a cell here. Okay, so the cell stops at the center, option number one, so it moves and stops. The cell continues down the gradient, so it crosses, as it does in 1D. The cell moves along the ridge. Option A, cell stops at the center. Good, you're listening to me. The cell continues down the gradient. The cell moves along the ridge. Good. All right, let's ask the cell to give us the answer. So the cell is slow, suspends, and then it starts migrating along the ridge. Okay, so all of you were right. I'm very happy. You have very clever people. Okay, good. So now this makes the problem more interesting, right? So the fact that we allow the cell to turn or not matters. So it's not just about polarity and adhesion, it's also about confinement, right? And this is important because cells that move in our body, they are not moving in a free space, right? So they are often confined, but sometimes they are not, right? So this is another variable that we need to take into account. All right, but then of course this begs the question, what happens when you're in between 1D and 2D, right? So we now make lines that are narrow, but not that narrow, no? So now the options are the cells behave as in 1D, so they continue to go. The cell behaves as in 2D, so it starts bouncing. 3, the cell does something really cool and unexpected. Okay, I will not ask you to raise hands. So here's what the cell does. It starts turning. It loops, right? So you turn some gradients in 1D, they start looping. Okay, so we have this phenomenology, right? 1D circles orthogonal trajectories, right? So then with our collaborators, we started scratching our heads, how we incorporate confinement to this system, how cells bounce on boundaries, and so on and so forth, and we were able to sort of capture that phenomenology fully. And so basically this means that you have 3 ingredients. 3 ingredients, one is adhesion, the other one is polarity, and the other one is confinement, right? And so you need to really take into account these 3, and then we have a model that is predictive, and then, so we submitted this, and the referees say, well, this is very nice, but it's very ad hoc. Can you provide us with more quizzes, but without touching the model? Is your model predictive of what will happen in other conditions that we're now testing? We don't have it ready, so I'm not showing to you, but maybe in the future I will be able to tell you about this. This is in the bioarchive, so if you're interested in reading this, you can check the paper. Okay, and I think I should be done. Do I have 5 more minutes, or do I just shut up?
It's not every day we have scientists like you here, so...
Okay. So I have 1 more hour.
5 more minutes we negotiate.
Yeah, 5 minutes. I just wanted to tell you a bit about shape, because I've been telling you a lot about migration. I wanted to tell you about shape, because this is something that we're investing lots of efforts in controlling cell shape. And I will not justify why shape is important. I think you all appreciate that if in the body we have shapes like this, we need to understand them. And within this context of very simple, sort of simple-minded bottom-up stuff, what we want to do is to generate shapes in which we can control them, we can control their mechanics, we can perfuse flows, and so on and so forth. So this is one of the efforts that we're trying to build in the lab. So we try to generate shapes from the bottom up, right? And then establish relationships between pressures, tensions, shapes, fates, and so on. And this is indeed 1 hour talk that I could give you just on this, but I will, I was not planning on doing this, but I will tell you just a few points. So we developed technology based on microfluidics, and this is work of these 3 people in the lab, that allow us to control shape. And the idea is very simple. It's very simple to draw this on a PowerPoint. It's hard to implement, but these guys are really good and they manage. So basically, we have like a transwell membrane. On top of that transwell membrane, we micropattern areas of high density and of low density. On top of that, we grow 1 monolayer of epithelial cells. Then we apply pressure through this sort of transwall membrane, and this pressure will delaminate cells from the area that is not adherent, but the cells that are on the area that is adherent, then they will stay stuck on this here, right? So again, easy to draw on a PowerPoint slide, hard to implement in the lab, but this works, right? So this is a top view, this is a side view, and you can see how we can now inflate and deflate this system. So we can sort of generate alveoli, if you wish, and we can control their shape, we can control their size, we can image it quite well. In fact, Thomas, his PhD has been really focusing on developing these tools so we can combine it with a light sheet microscope, so we can now obtain sort of beautiful images of these domes. We can see how they are growing, how they are shrinking. So we can really make 3D segmentations of these cells. We can look at the nuclei of each one of these cells in 3D. So we can really ask questions as how this system is actually deforming, relaxing, and so on and so forth. I just wanted to tell you about some really surprising findings that Tom goal they found. Tom was interested in asking the question, okay, how does the cell withstand this stretch, right? Because inflating a cell like this means that the cell is doubling, tripling, quadrupling its area, right? If I quadruple the area of my jacket, I will break it. Cells quadruple their areas all the time in our body, and they never break. So how do they manage? And so to address this question, what Tom did is to look at actin, which is the main candidate. He looked at actin, nothing happens to actin. It doesn't play a role here. He looked at intermediate filaments. And intermediate filaments, he found that there is a very drastic topological transition in their structure. So if you look at cells that are not deformed, cells that are in the monolayer here, you see that intermediate filaments and keratin in particular, they are distributed around the nucleus. They form a cage around the nucleus, like a protective cage around the nucleus, right? And then they fill the cytoplasm. But when you stretch the cells, the intermediate filaments adopt this shape, or the shape that you're seeing here, right? They form these bundles of filaments that connect adjacent cells. And then you have cells that in part are connected to these bundles, and in other part they are not, right? So you have these three topologies as you're stretching the cell that these cells are adopting. And we were really interested in understanding how this drastic topological transition takes place in the system. So for that, we use this system where we inflate the cells, we hold them inflated, we look at what happens. And so when Tom did the experiment, he inflated the cells, he looked at what happens, and the answer is, Xavi, nothing happens. And then this took like 4 years to us, because we know that it happens, because we have stainings showing that it happens. Tom does the experiment, nothing happens, nothing happens. And finally, Tom managed to do the experiment for a very, very, very long time, and then he found that indeed, when he waits for long enough, many, many, many hours under the microscope, this is very hard experiments, he does see that these cells end up bundling. So I'll play this movie again, because it took us 4 years to get it. So these cells undergo this bundling transition, they make this star-shaped structure. So we believe that this transition is actually what helps these cells to withstand very large stretches, because they undergo this transition, and then this provides them with a more resilient structure. So here's a zoom of what this bonding process happens, right? So you start with nothing, you end up with bundles. And the way this works, you can understand it if you start looking at one tricellular junction. So you look at the tricellular junctions, intermediate filaments are located around it, but with time you will see that intermediate filaments, they become depleted from this tricellular junction. They make this sort of bubble, an area that is depleted of intermediate filaments. So how does this happen? So we did what we do when we don't understand something, which is we talk to theorists. And these theorists, Marino and Marco, they developed a model. And this is a very simple model. It has just 3 features. You have an hexagonal cell, entangled filaments that are connected at one edge of the cell and to the other edge of the cell. They can slide along the junctions. And then what they did is they start with this configuration. It's like a spaghetti soup. You stretch it, and then what you get naturally is bundling. Okay? So, yeah, I think my simulation was stuck, but okay. It's smoother, but at the end what you get is a central knot, right? So you really get, just out of geometry, this is not even physics. This is not physics, this is mathematics. Okay? It has to be like this, right? So you form a knot at the center. And then, but then, so this was satisfying, right? Because just one simulation allowed us to understand something that we had really been struggling to understand. But then this raised many questions, and one of these questions was, okay, in our simulation there's no nucleus, and we know that these keratins are actually wrapped around the nucleus, right? So the question was, what happens to the nucleus? And to understand what happens to the nucleus, I will give you like the simplest mechanics class that I can give to like kindergarten students. So you have a rope and you pull on it, the rope will be under tension. But if you have a rope, you make a knot, and you pull on it, then you will be able to generate compression at the center. So you generate compression by tension. This is what you do every morning when you tie your shoelaces. Okay. So what seems to happen, or our prediction was that, okay, if you're pulling on these filaments and there's a nucleus here, then you will sort of generate a pushing force. And this is weird mechanically, right? I'm pulling, but the nucleus is pushed, right? It is compressed, right? So to convince us that this made sense, now they did a 3D simulation, and this is very complicated. But basically what they found is that when you stretch this keratin network, it's actually compressing the nucleus, and eventually, if you pull hard enough and you pull long enough, the dynamics of the system are so that the nucleus will escape this keratin cage, and eventually you will have this cluster of filaments underneath the nucleus. So the nucleus escapes its protective cage, right? I promised to you that they made these simulations before we tried the experiment. We had never looked at the nucleus. So, but Tom looked at the nucleus. And so this is my last slide, where you can see at the beginning of the experiment, you have here the nucleus, you have the keratin filaments around the nucleus. In this side view, you can see this is within this cage, right? So you have keratin here, you have keratin here. So the nucleus is really protected by these keratins. We stretch for a few hours, nothing happens. We stretch longer, and then we find that the nucleus is now sitting on top of keratin. And if you wait even longer, and this stays, and it's actually irreversible. So to these sort of topological transitions, not only the cell has a mechanism to probably adapt to stretch, to support larger stretches, but this also has implications in terms of the nucleus escaping. And this probably has implications in many, many other sort of regulatory processes, because we're now uncoupling two key structural elements in the system, one of them that contains all the genome, of course. So that's something that we will continue, or maybe Tom will continue to study in the future. All right. So with this, I will just finish with, this is a picture of my group a couple of years ago. I showed names and faces of people who did the work. I wanted to talk about collaborators, because this at the end is, I'm here because of an award, and the award happens just because these people are amazing, and because these collaborators are also amazing. So collaborated with Marino Arroyo and his group on the theory side, with Ricard Alert and his group on the theory side, as well as with Jaume Casademont. So we collaborate with theories. These people have bigger brains than us. They can really think in a different way than us. And then collaborated at Curie for organoids, Eduard Batlle for our work on cancer that I didn't talk about, and also other theorists at IST. These are our funders. We're grateful to them as well, and I thank you for your attention.
Xavier, thank you so much for this fascinating talk. And I think it illustrates very well the importance, as you concluded here with your slide, of addressing very basic biological questions with an interdisciplinary approach. And I'm sure we're all very grateful that you oriented the content of your talk today more towards an audience where we are biologists rather than physicists, because I'm sure you could have done it from either of those angles. And also the work that you're doing, that you've done over your career, certainly illustrates the importance of creativity and of having fun. And I think those two elements really feed off of each other. That when you have fun, you're much more likely to be creative, and vice versa, I think. So we can open the talk now for questions for Xavi.
Now you can do the quiz to me.
We have several group leaders that you would really enjoy having a beer with sometimes, because they spend a lot of their time, free time, thinking about very basic questions like you do. And some of them are here on this row. Luke Noon, Iborda are people you'd really enjoy sharing a night of beer.
I can come back. I mean,
I don't drink, so I haven't for a day at least. Can I just ask about your experiments where you have the chains of cells? Have you it's a gain of function experiment, but what happens with the loss of, did you do loss of function experiments? Did you ablate the cell, or did you?
In cell migration, you mean, in the chains? So, I mean, these cells, we spontaneously, some of them are just don't move when they are in these doublets. Some of them move spontaneously. So we have not made them move less, because we really wanted to control the onset of movement. There are other things that we could do, like, for example, increasing their contractility with light, which would go a bit in that direction that you're saying, right? If you make them so less RAG1 will make them more raw, more contractility, maybe other modes of migration. So I think there's stuff to explore in that direction, but like a specific I mean, we're developing lots of optogenetic tools, and we're developing some sort of RAG gaps that should do that, but we have not done it.
And how would you expect your chains of cells to behave on the gradient of fibronectin?
Yeah, and there's these questions about fights, right? So you want to move up, and I want you to move down, so I will photoactivate you in the other direction, no? And so these are fun experiments to do. We have not done it, but I would say, I mean, one thing we learn in this system is that polarity is a very strong cue, right? Especially when it comes with confinement. No? That, I mean, these cells can go on down a gradient, which is a very strong negative signal for a long time. No? So whether we would be able to reverse that with light by like forcing RAG1, no? We haven't tried it, and it's one of these sort of Friday evening experiments that I would have done, but no one has done it in the lab yet.
Thank you very much. Very nice talk.
Thanks.
If we think in a tumor tissue that it's a disorganized tissue, no, compared to a wild-type tissue, migration follows the same rules.
So in a tumor, I mean, there's one very important thing in tumors is that it's very packed, right? A tumor is very packed. So cells must migrate within very narrow spaces. And therefore, this idea of confinement, of course, not in the idealized confinement that we make, but the idea that a cell doesn't have a 2D space to explore, it will have to migrate through very narrow channels. And one thing that we learn is that in tumors, cells that are either metastasizing or immune cells that are sort of squeezing in, so this polarity cue is very important, and it can actually override many other signals, right? So if there's something that makes, I mean, one question that we always have is why cells escape the tumor during invasion, right? Because this seems to go against mechanical gradients, and why are they not staying in the stiffest, more dense? And so one possibility is that really these signals might be like overwritten by some sort of polarity cue that is acquired of some sort. And once the cell is polarized, it's actually hard to sort of change that, no? And if you think about it physically, like if you have all your machinery on one side, like moving it all to the other side, this is a very unlikely event, no? So, yeah, but we don't know exactly what happens. But there's also all these amoeboid migration modes that happen in tumors. Everything I showed you is like mesenchymal, and these cells in tumors, immune cells certainly. Also, many metastatic cells, they are amoeboid, right? Blabbing and so on. So it's a completely different mode of migration, different from what I told you about today.
Hi, really nice talk. Thanks a lot. I'm going to do this. I have more of a comment than a question thing. So regarding your really cool experiment with the photo activation, my concern is that you are just activating the movement, but sometimes when this collective migration happens, the leader has many more functions than just moving. So, and they always interact with the adjacent cells to promote some other things there. Mm-hmm. So I'm wondering, are you thinking on investigating this in this direction?
Yeah, I mean, this is a really good question. Because it helps me clarify what we're trying to do. And I mean, so we're not disputing the idea that there are leader cells in nature, because we know there are leader cells in nature, right? And in development. And our question is really like, a mechanical leader is enough, right? And the answer is no. But what you're saying is that, I mean, cells are not just mechanical agents, right? So they can establish all sorts of communication strategies. And so what our work tells you is that whatever these strategies are, if the cell wants to move as a group, there will have to be so that a mechanical asymmetry needs to build up. Now, you're completely right that, and we don't know, and we didn't address, what are these communication strategies that allow the cell to do that. But certainly, from the moment that we observe in nature this sort of leader-follower organization, what this means necessarily that there are mechanisms in place, many of which will involve sort of signaling and ERK and waves and so on, that will allow the system to move as a whole. No? But there's a lot of literature that just makes the point that all you have is like a train, you know. So you have an engine, the locomotive, and then you have the wagons, active and passive. No? And this idea is in the literature since the beginning of the 20th century. No? And I think we killed this idea with this paper. And we open another question, which is that then, what are the mechanisms that allow the system to communicate so that it establishes robustly this asymmetry, this mechanical asymmetry?
It's cool, and there is also a philosophical debate there that you need more than just pulling to be a leader. You need to ask them to push and things like that. It's really cool. Another question would be on the last part. How is the transcription in this compressed nucleus? Have you checked if it is induced or inhibited, or how is it working?
Yeah, so this is a question that I have asked Tom to do. Transcription, DNA damage, and he's like, no way, I'm not looking at this. So we will let referees ask for this question, because I think it's super important, right? So you have a nucleus that is in a cage, and now it escapes. To escape, it squeezes out. I'm sure this will have consequences for transcription at all levels, right? And DNA damage and whatever. We'll look into that. It's not, I mean, these are hard experiments, but it's necessary to know this. It's necessary to know this. And I'm sure there will be, and we'll look into those. One thing we know is that when so I told you an experiment where we inflate and we wait. So, and then these bundles from, when we relax, all the cells that have bundled, they die. They are extruded. I don't know if they die, but they are extruded. So it's not something, it's irreversible. So it's a program or whatever, however you want to call it, it's a strategy that has evolved in a way that these cells are able to sustain very large stress, but as soon as stress is released, these cells have done their job. And they are probably not viable for everything else, and then they are extruded, they are gone. And then the epithelium reforms, and you're back to start. Yeah.
Yeah. So congratulations, amazing talk.
Thanks.
To the quiz, I answered that the cell would have continued, and I guess so because, to my understanding, the adhesive structure, apart from being adhesive, of course, they are also very complex and dynamic signaling platform. And to my knowledge, they can go through different developmental stages, they can mature, and at least in the growth cone, they normally have an orientation. So I would have predicted that they would keep a sort of signaling inertia in terms of molecular composition and phosphorylation state. So
you mean in terms
I was
of
wondering whether you could look at these kind of things in your system, like with Paxillin GFP or some other markers for maturation.
Yeah, I totally agree. So I mean, there's all these dynamics at the leading edge, these, focal adhesions, there's this wave complex, all this signaling machinery that is established, this polarity machinery, in fact, that in a system like this you can study it very, very well, very systematically, and we really we have done a lot of more focal adhesions. We've looked at focal adhesions, we've looked at retrograde flows, at actin myosin localizations. I didn't tell you about it, but yeah, I think there's a lot of these. I think there's a very important question of how this goes away, right? Because at the end, there will be a moment where the cell will turn, right? Not as soon as we would have expected, maybe, but so there's a very interesting question of all this polarity signaling that you are talking about. At some point, it is dominated by adhesion signaling, and then the cells reverse. So this system allows us to study this very well, right? Very quantitative, because you can visualize all this signaling with FRET sensors, with whatever, and then you can really study very carefully this moment where this dominates until a moment where the thing changes, right? So, and I agree that this is very interesting. We're not experts in this. I don't think this is something that I would be able to do in a sort of impactful way. But I think it's a very interesting question, yeah.
More questions? Moving away from this row, the microphone is following the same kind of pattern of migration of being pulled along.
Hi, thank you so much. Yeah, I really enjoyed the talk. So I have a question regarding the migration of fibronectin gradient. So as you know, like fibronectin has binding sites for growth factors. So how are you sure, like, I was just wondering, how are you sure that there is no, like, a gradient as well of growth factors coming from maybe the media?
Yeah.
Not like a chemotaxis or?
Yes. So it could be. We have not ruled out that. This is something that people still call it apoptosis if it's immobilized. They don't call it chemotaxis, but it's a different mechanism, right? It is. So it could be. It could be. I mean, and I don't know how we could rule this out. We could sort of flash things, but I mean, I don't know. Can you think of a way that I could use to rule that out?
Will be hard because I mean, like using a fibronectin that doesn't have the binding site maybe will be an option.
Yeah.
For some of the growth factors, there are mutated fibronectins. So
Yeah.
maybe,
Yeah.
I don't know. Will be just the conformation will be maybe different as well. But yeah. Anyway, I was just curious to know. Maybe without media, without any serum, I don't know how long.
Yeah, there's very little serum here, but there is some. And yeah, I mean, one thing we're careful with is that we don't let the cell pass on top of the lane. So we seed the cells and the matrix is bare because we know that when the cell migrates, it drops a lot of stuff behind it. And maybe in a gradient as well, as you're saying, right? But whether there's a gradient, we could print gradients of growth factors that we could do, and maybe we can do this like maybe homogeneous fibronectin, and then we make a gradient of whatever growth factor, and we see if this is sufficient. No? But to rule it out would be complicated, and I'm very glad that you did not review our paper.
Thank you. Thank you so much.
I wanted to ask you about the asymmetry traction, this concept.
Yeah.
So I realize that you've defined it in a particular cell system with a particular type of cell. But my question is about how the environment in that particular system determines asymmetry traction, or does asymmetry traction define the system? Because if you change the system, for example, rather than having the cells on plastic from Corning, you changed them to plastic from Fisher. Or you plated them on gelatin. How would those modifications to the system change the asymmetry traction?
Yeah. I mean, that we don't know because, I mean, so far what we've done is everything I showed you. But, and then, so how this asymmetry is established, we don't...
That's what I'm asking.
Yeah. And so how the cues, like how uniform cues, so now we know how sort of gradients in a way establish this asymmetry, but how they might arise spontaneously, right? I mean, they could arise stochastically. Like one thing we do is when we did in this homogeneous system without illumination, so we don't do anything. Some clusters move, others don't. We look at them and they show the same asymmetries than when we induce with light, right? So there is some ways in which symmetry is broken in the system, and then these cells start migrating. And it could be as simple as you have a doublet, and then one cell is more contractile than the other, and then the symmetry is already broken, right? So maybe it's just very small fluctuations in one component are sufficient to. I mean, these systems are mechanical and stable, right? In the sense that they are subjected to these positive feedback loops that make the system break symmetry easily. So, but again, that's going back to that question that this involves, I think, some communication that is not just mechanical, and we don't know. I mean, the answer is we don't know, and as I said, I mean, I think that many of these questions are not for us to address because I don't think we have the know-how.
this is my final comment today. Probably only the group leaders, or some of the group leaders will know this, but we had proposed to EBEC, to the director at EBEC, and Anna was actually working in this direction, that we would organize a retreat, a joint meeting between EBEC and our center, because I think we have very complementary perspectives about basic biology. And I'm sure there are many groups in the audience that would be very happy to collaborate with you in some of these questions, because when you show, for example, this fascinating video, and I could actually look at it all day long in the beginning, about this patch of cells closing the gap of healing, or whatever you want to call it in that context. And so